Welcome to this video. This video is going to be all about the digestive system. Everything that your anatomy teacher might ask you on a test about the digestive system chapter in your textbook. We're going to start off with an overview of the digestive system, its processes, and its organs. Now, if you go into a healthcare field, you're typically going to hear this called the gastrointestinal system or the GI system, and that's the same thing. Okay, so you can see from my sketch here, I have the organs of the digestive system. Now, some of these are considered accessory organs, and some are actually part of what's called the alimentary canal. Alimentary literally means food, so it's probably no surprise that this is going to be the organs that the food actually passes through. Now, we also have accessory organs. These are ones that the food is generally not touching as much. There are some exceptions, like in the mouth, but these are organs that typically are going to be important in making different kinds of secretions for digesting and absorbing the foods that we eat. So let's go ahead and we'll use kind of like a red-pink for all of the alimentary canal organs, and we will use, I guess, blue for all of the accessory organs. So let's start off with the oral cavity. I'm just going to put a box around it. So this is a little sketch of a mouth with some salivary glands and things like that. So we'll put a number 1 on here. And this is called the oral cavity. Oops. Now there are some what we call accessory organs within that area. And these are things like salivary glands. See these? We have three different kinds of salivary glands. Teeth, there's some teeth, and the tongue. So these are considered accessory organs. So we'll put teeth, tongue, and salivary glands. These are primarily going to mix up the food to form what's called a bolus or a ball that can then be pushed down to the stomach. So the next place that the food goes is into the throat. We can maybe just put a circle here, I guess. And that is the pharynx, is the fancy word for it. But this is just what you know as your throat. Now, by this time, the food has formed into a little ball that's been all mushed up, and we call that a bolus, which literally, you can see right there, right? It means a little ball of food that can be pushed down into the esophagus when you swallow. And the esophagus is this long tube. It's about 1 foot long, and it is going to get the food, get the bolus into the stomach, which would be the next structure. The stomach is going to do a lot of churning, a lot of what we call mechanical digestion, where it just makes the food get smaller and smaller. Then the next structure is the small intestine. So I'm just going to put a 5 here for small intestine, and then I'm going to show you the 3 parts of the small intestine. The first part, I'll go ahead and color like this. This is the first curve of the small intestine. It's only about 12 inches long, and it's called the duodenum. The duodenum. Some people call it duodenum, and I have looked, and I really do find that people say it both ways, and I think it's fine, whatever you like better. Okay, now the next section It's actually much longer than I'm putting in my sketch here, but that's the jejunum. The jejunum. And that's where most of the absorption finally occurs. Then the last section of the small intestine is called the ileum, and it ends up at the large intestine. So that's the ileum. Which brings us to, so here I can put like this. So I guess for the jejunum we did solid, right? And then for the jejunum we did slashes, and for the ileum I did dots. How's that? And the stomach was also dots, wasn't it? And the esophagus was slashes, and the pharynx was solid. There. Okay, so then the last structure would be the large intestine. You're going to often hear this called the colon. Like if you've heard of colon cancer, it's cancer in this area of the body. And it starts with what everyone's heard of, the appendix, and that's attached to the cecum. So this is the entry point. So we'll do a solid, and we'll do cecum and appendix. And then the whatever's left over at this point is going to end up in the feces except for water. So the food is passed up the ascending colon, across the top, which is the transverse, down the descending, around the bend of the sigmoid colon, and then finally when it straightens out right before the anus, it's the rectum. So we go ascending colon, transverse colon, descending colon, sigmoid, and then finally to the rectum. Those are going to be all the portions of the large intestine. Over back on the accessory glands, when the food enters the small intestine, it gets squirted on with a bunch of important enzymes. So the pancreas makes most of your digestive enzymes. I'll just go ahead and color this. See how it kind of sits in that first curve of the small intestine, and then it has an ability to secrete its enzymes right into the duodenum. Isn't that cool? So that's going to be the pancreas, makes most of the enzymes that you need to digest your food. And then look here, we've got the liver. The liver is going to make a substance called bile that will help break up fat droplets into little droplets. It's a form of mechanical digestion. It's not actually changing the chemical composition of the fat yet. You need pancreatic lipase to do that. So the liver, so the pancreas I colored solid, right? And then the liver, we put some hashes like that. So the liver makes bile. But the gallbladder, this little guy right here, we can do this one solid too. This structure, it's actually on the underside of the liver. The gallbladder stores and concentrates the bile. Let's look at the oral cavity, the parts of it, and then the structure of a tooth. We will start by coloring the lips red on this mouth over here. So these are the lips. And then we can make the gum line kind of a softer red, almost like a pink. So this is also called the gingiva. So we'll call that the gingiva. And then the palate is the roof of the mouth. And hanging from the palate is, the back of the palate is a little structure called the uvula. You can see it when you look in the mirror. and it hangs down at the back of your mouth. And its job is to move upward and protect your nasal passages from food entering them when you swallow. Then I'm going to use yellow and color these tonsils at the back of the throat. These are the palatine tonsils. If you've ever had strep throat, these are what get the white patches on them. And they're meant to guard the entrance to your digestive system from pathogens that are trying to enter. Then there's the tongue right here. Tongue. And the tongue is anchored to the floor of your mouth with something called the frenulum. It's a little strip of connective tissue that connects the tongue to the bottom of your mouth. Now we can look at the teeth. So when you are little, you get 20 baby teeth. So babies get 20 deciduous teeth. Now they're called deciduous because they fall out. That word deciduous means fall off, and that's why trees that lose their leaves in the winter are called deciduous trees. These baby teeth start coming in at about 6 months of age. And then they get all of their 20 deciduous teeth. And then when they're about 5 or 6, those start falling out, and then their adult teeth come in. And my boys, they started getting their adult teeth when they were about 5. I guess that's when their first tooth fell out, but they think they were a little bit on the young side. But they come in about 6 years, and then some people will even get a third set of molars by the time they're like 21, but I'll put about 18 for that last set. So speaking of molars, let's go ahead and look at the 32 teeth that an adult could have. So you first got what are called incisors. Here's one and here's one. So these are called the incisors, and their job is to cut the food. These are the first ones that little babies get. And then there is that real sharp tearing one called the canine, one on either side. And this is going to tear the food. And then there are a couple of what are called premolars in the textbook that my students are currently using. But you'll also see these called bicuspids. And then the last 3 are called the molars. And the very last one is also called the wisdom tooth. Not everyone will get those last ones, and some people's mouths can't really even fit them, and so then they'll get them surgically removed. So the molars, their job is to grind the food up. Next, let's look at the structure of a tooth. So over here, you can see that I've started a little drawing of a tooth, and the part that is visible above the gum line is called the crown of the tooth. So this is the part that's above the gum line. And I'll color the gum line here for you so that you can see what I'm talking about. So this is the gum line right here with that gingiva, remember? So the part of the tooth that's above, that's the crown. And then the part where the gum line is called the neck of the tooth. And then the part that is not visible above the gum line is called the root. So this is below the gum line. Now, the tooth is protected by very strong mineralized structures. So mineralization protects the tooth, and I'm going to show you the 3 types of mineralization in the tooth. And that mineralization, it's basically like turns part of the tooth into a rock, basically, is made of calcium, hydroxyapatite, crystals. Now, these are the same, similar kind of structure that we find in a bone. It's just that a bone has a lot more collagen fibers in it to give it flexibility. This mineralization makes the tooth very, very hard, harder than bone. So we'll start with the top layer that is called enamel. You've probably heard of that. The hardest structure in your body, and it's basically 100% mineralized. So it's like a rock. And then below that, and that's going to be covering the crown of the tooth. And then below that, we're going to have the dentin. I'm going to color the dentin, which is a little bit softer than enamel. I'm going to put that here. So this is below the enamel. And I'm making it all in blue. This is called dentin. So we'll put dentin here. And that's going to be about 70% mineralized. So do you see that it's softer than enamel, but still a hard tissue that helps to protect us? And then the other parts that aren't calcium hydroxyapatite would be proteins and things like that. Okay, then there is one more layer of mineralization protection, and that's right here. Do you see basically along the root? And I'm making this in green. And this is also hard. This is called cementin. And cementin is about 50% calcium hydroxyapatite. So let's put cementin here. So it's 50% mineralized, and that is the green. Maybe I'll put a green line right here, and then a blue line right here, and then the enamel is on the white top part of the tooth. All right, so now once you understand that important protection for the tooth is provided by mineralization, let's look and see what's so special on the inside of the tooth, and that is in the pulp cavity where we have the blood vessels and the nerves. So I'm just going to put these lines right here. Do you see how those actually go out the bottom of the tooth? So this is where the blood supply for the tooth can both enter and exit. So I'm putting red and blue lines to represent arteries, or actually capillaries and venules and things like that, small arterioles. So we have blood vessels. And then I'm going to use this yellow because you have a lot of nerve endings in here too. That's why a cavity can hurt, or if the tooth starts to erode, then the nerve endings here are very sensitive. And we call that area the pulp cavity. So the pulp cavity is vulnerable because it is basically 0% mineralized. It's very soft. And so I can go like this to remind you that this is the pulp cavity. Like that. And put an arrow right here to that pulp cavity. So this is where we're going to find the blood vessels and the nerves. For the tooth. The teeth fit into the mandible and the maxilla in what are called alveolar sockets, and so the mandible and the maxilla, so that's your lower jaw and your upper jaw. And they have alveolar sockets for each tooth that forms an immovable gomphosis joint. And that's something you would have learned about when you studied the skeletal system. Periodontal ligaments hold the tooth to the bone. And these ligaments, like all ligaments, made out of collagen fibers. So you can see these collagen fibers are anchoring the tooth to the surrounding mandible and maxilla bone within the alveolar socket. So these are the periodontal ligaments. They anchor the teeth. So maybe you've heard of scurvy when someone doesn't have enough vitamin C, because vitamin C is necessary for the production of collagen. And the sailors, if they got scurvy, their teeth would become loose in the sockets. We'll look at the salivary glands on this page. You have 3 sets of salivary glands. This first one I'm coloring is called the parotid gland. Name makes sense because it means beside the ear. Parotid. Right here. And it has a duct that releases saliva into the top of your mouth, kind of back by your second molar. And so then that saliva will drip into your mouth and be mixed with saliva coming from the bottom of your mouth and moved around by your tongue, which is right here. Oh, I was going to put, this means beside the ear. That's where it gets its name. And then you have the submandibular gland, and it gets its name because it's under the mandible. Or below. And we'll make this one also yellow right here. Do you see it? One of those on each side of your mouth. And it is going to release its saliva here. And then you have under your tongue a salivary gland called the sublingual, which is another name that makes perfect sense. Sublingual means under the tongue. Now this one has sometimes 8 to 20 different ducts that go out into the bottom of your mouth. So you have lots of different places where saliva is entering your mouth. These salivary glands are controlled by cranial nerves from the parasympathetic nervous system. So parasympathetic control over salivation for stimulating it. The parasympathetic nervous system is part of your rest and digest, so it's going to help you make saliva so that you can start to break down your food. And there are 2 cranial nerves. There's cranial nerve number 7, which is the facial, and cranial nerve number 9, which is the glossopharyngeal. That means tongue and throat. So those are the 2 nerves that are most associated with stimulating salivation. It contains an enzyme called salivary amylase. Now, the reason we say salivary, it seems kind of obvious, right? You make this enzyme amylase in your pancreas too. The job of it in the saliva, as it is also in the small intestine, is that it begins the breakdown of starch, which is a carbohydrate. So starch digestion begins in your mouth. And this is a carb, if you had forgotten. That's a carbohydrate. And saliva also contains another enzyme called lysozyme. This enzyme is basically found in all bodily secretions, and it's able to kill bacteria by damaging cell walls. There's another really neat ingredient found in saliva, and it's a specialized kind of antibody called IgA. That stands for immunoglobulin A. And this particular kind of antibody is specialized for hanging out on your mucous membranes and defending your body against foodborne illness and things that might come in on your respiratory tract as well. So this is an antibody that's specialized for defending your mucous membranes. Oop, ran out of room there. Just defending mucous membranes. Now, this saliva, there's also going to be water and mucus in it and some electrolytes as well. It's going to get mixed up as you chew. So I'm going to take a red, kind of a pale red, and color these two chewing muscles. So the technical term that we use for chewing is mastication, and your two most important chewing muscles are your temporalis and your masseter. So right there, I just drew the temporalis. So mastication includes the temporalis, which I've colored. I think I'll use black here to show you. So that's the temporalis, and then also the masseter. Here we go, color this. This is the masseter. You can see how both of these, based on where they insert, can close the jaw. Right there, you can see how both of those pull the jaw closed. Mastication means chewing, and then once you've chewed up the food, you have mechanically digested it, meaning you break it into smaller pieces, and thanks to the salivary amylase, you've begun a chemical, I should put a chemical breakdown, so it's a chemical digestion, and now you're ready to swallow. And I'll put that word right here, which the fancy word we use is deglutition. And this word means to swallow. And we swallow a bolus, which just means ball of mashed up food. And then that's going to go down the esophagus and into the stomach, and we'll look at that next. So it's going to produce this thick, uniformly mixed up substance called chyme, and this chyme is going to end up being very acidic, and then it's going to go down into the small intestine, and then those substances will be chemically digested the rest of the way. Let me show you how this works. First of all, you've got the esophagus here that's coming down from the pharynx, and the esophagus uses a process called peristalsis, which is a rhythmic contraction to push the food along the alimentary canal. And when the food arrives at the diaphragm, the esophagus actually makes a hole through it. I'm going to highlight this. This right here is a skeletal muscle called the diaphragm. It's very important in breathing. So just under the diaphragm is where the stomach begins, and it has a circular or sphincter-shaped muscle that guards its entrance and its exit. So see how there's a circular muscle right here, and then there's also one that guards the exit to the stomach. And that way the stomach can make sure that it mixes everything up uniformly and gets it to the right pH before it allows it to leave. This top sphincter is called the lower esophageal sphincter. Sphincters are always circular-shaped, smooth muscles, and sometimes it's called the LES. Now, a lot of textbooks will call this the gastroesophageal sphincter, but clinically you're going to be hearing it called the lower esophageal sphincter much more. And it's going to prevent regurgitation of acidic chyme. The esophagus is not designed to deal with that acid the way that the stomach is. So if someone does have a problem and this is happening when it shouldn't be for a variety of reasons, then they might have gastroesophageal reflux disease or GERD. Okay, then you also have a guard at the end, and this one is called the pyloric sphincter. That word pyloric means gateway or gatekeeper. And so this is going to guard the small intestine. So this is going to prevent chyme from leaving before it's good and mixed. And the pH is very low. It's about 2, which is neutral is 7, so that's super acidic. Okay, so you see how we have these two guardrails? Okay, now I've been telling you how it mixes it up, right? So let's use a red, and let me show you, just using some lines, that the stomach has 3 layers of muscle. Sort of like our abdominal muscles, we have the rectus abdominis, the internal obliques, the external obliques. We actually have the transverse abdominis too. Well, similarly, the stomach has layers upon layers of muscle. Some of them, the fibers run longitudinally, like this. So we call these longitudinal layers. It's a longitudinal layer. And then there is a layer that runs around the stomach like this. And we call this the circular layer. And then there is even a third layer that runs at an angle like this. And just like on our abdominal area, we call these angular ones the oblique. So this is the oblique layer. Pretty cool, huh? So you have 1, 2, 3 layers of smooth muscle on the stomach, and then when they contract, they can make the stomach get shorter or wider or angled, and that's gonna make sure to get it all mixed up into that uniform chyme. See how the stomach has a curve right here? We call that the lesser curvature. And there is a mesentery that attaches to this lesser curvature. And so we call that the lesser omentum. Let's color that right here. So this actually attaches up to the liver from the stomach wall. And this mesentery helps hold things in place and also is a place for blood vessels as well. So this is the lesser omentum. You can maybe see where I'm going with this because we're also going to have one that's greater. Oh, and that word omentum means apron. So it's like an apron that covers this top part of the stomach coming down from the liver. Bigger curve of the stomach. So we call that the greater curvature. And coming down from the greater curvature is the greater omentum. And so this is another mesentery. This one's really good at getting filled with fat. So as we get overweight, we can store a lot of fat in this greater omentum. This apron-like mesentery hangs down over your intestines. So this is the greater omentum or apron. Next, I'll name the different parts of the stomach. So the very entry point that's right near the heart is called the cardia of the stomach. And then there's a little part that actually, my picture doesn't show this very well, but it's called the fundus. And this is the part of the stomach that protrudes above the opening, which I didn't really show very well in my picture, but the fundus goes up above the lower esophageal sphincter. And then the bulk of the stomach is all called the body. That's going to be most of it, right? And then this part that is the inferior portion is called the pyloric antrum. And then the last part is called the pylorus, and that's where we find the pyloric sphincter. Regions. You've got the cardia, which is the entry point. Then you've got the fundus, which is the highest portion. Then the body, that's most of it. I'll put the bulk. Then the antrum, it's the pyloric antrum. That's the lowest part. It's almost like opposite to the fundus in a way. And then you've got the pylorus, which is the exit point. So it's opposite of the cardia in a way, right? Cardia is the entry point, pylorus is the exit point. If you were to cut your stomach open and look inside when it didn't have food in it, you would see a lot of wrinkly ridges called rugae. And what these rugae do is they allow the stomach to be able to distend, because as the stomach fills up, then it can get bigger and bigger, and those rugae sort of flatten out. This is why we can really eat a big meal, even though we might feel a little bit overly full afterward, right? So these are the rugae. I want to describe to you the classic phases of digestion. The first one is called the cephalic phase of digestion. That word cephalic means head, so this one happens before you even start eating. The stimuli for this first phase, since it's all in our head, right, is going to be the sight of food, the smell of food, and even just thinking about food. And then what happens is our parasympathetic nervous system kicks in and sends signals down the vagus nerves. The vagus, or cranial nerve number 10, is known as the wanderer. And what this is going to do is start causing your stomach cells to make stomach acid already. So we have cells called parietal cells in our stomach wall. That word parietal means wall because these cells are in the wall of the stomach. I think I'll do it like this. So this represents a cell in the wall of the stomach, and this cranial nerve stimulates this kind of cell to make acid. We'll put it like this. It actually releases hydrogen ions, and then those mix together with chlorine in your stomach and combine to form hydrochloric acid. So your stomach is getting ready for the food to arrive. It's already producing acid. To actually help out even further, your cranial nerve will stimulate another kind of cell in your stomach wall, and this one is called a G cell. It's called a G cell because it makes a hormone that will travel in your bloodstream, and that hormone is called gastrin. And that gastrin is going to also stimulate the parietal cell. So do you see how we have multiple stimuli for those cells to keep producing hydrogen ions? And then they can combine with chlorine to make hydrochloric acid. This is why just thinking about having a really good meal, if you think you're going to get it and then something happens to delay it, you might actually have a little bit of acid indigestion because your stomach was producing acid in anticipation of that delicious meal. Then we come to the gastric phase of digestion. And this one is going to be what happens when the food actually enters the stomach. So the stimuli is a bolus of food, and especially protein. It's going to be the best at stimulating this gastric phase. There's another kind of cell lining the stomach wall called a chief cell. And this chief cell is going to release an inactive precursor to an enzyme that's called pepsinogen. Pepsinogen. Now, whenever you see ogen at the end of something in anatomy and physiology, it means that that molecule is inactive. It's not doing its job yet. It's just waiting in the wings. And what it's waiting for is acid. And so it can be converted into its active form called pepsin. See how we took the O-gen off the end? And the conversion of this is hydrochloric acid. It makes it happen. So we can go from an inactive pepsinogen to the active enzyme pepsin. And then what do we need that pepsin for? Well, that word pepsin means protein. Pepsin is going to convert the protein that's in that food that enters the stomach, and it's going to break it down into smaller peptides. So it gets the chemical digestion of protein started. We have to have acid for that. So if you don't make enough stomach acid, or you take too many stomach acid blockers, you might actually have trouble breaking down the protein in your diet. One other thing that acid is really important for is not just that it's going to help us chemically digest protein by activating pepsinogen to pepsin, but it also is good at killing bacteria. So we can say that acid has two important jobs in our digestive activities. One is to keep us safe from foodborne illness, and the other is to begin the chemical digestion of protein. By the way, parietal cells secrete one other substance, and it is called intrinsic factor. And intrinsic factor is necessary for the absorption of vitamin B12. I'm going to put this over here since I don't quite have room where I'm talking about it. So intrinsic factor is necessary for vitamin B12 absorption. And if you have a deficiency in vitamin or in intrinsic factor, then you're going to not have enough vitamin B12, and the person will end up with something called pernicious anemia, because vitamin B12 is necessary for red blood cell production. We will talk a little more about that later, but I did want to put it on here to get us started. Then we come to the third phase, the intestinal phase of digestion. And the stimuli for this one is the acidic chyme, that's the mixed up stuff from the stomach, that enters into the duodenum, which is the first part of the small intestine. So acidic chyme entering the duodenum. So now we have two more special cells come into play, and both of these cells are in and part of the intestinal wall. The stomach lining is specially designed to deal with acid, but the small intestine isn't. So it's important that we have a way to inhibit too much acid in the small intestine, because it will get damaged. So these cells in the duodenum wall will make secretin, and secretin is going to inhibit. See how I make that straight line? That means inhibit. So secretin will inhibit further acid production from the parietal cells. Do you see how my picture shows that? So that's one job. But then it's going to do something else to help the duodenal wall keep from getting burnt up, and that is that it's going to stimulate the pancreas to make bicarbonate, which is, like baking soda, and it will help to make the pH become more neutral in the duodenum so that the enzymes will be able to work in the intestine and not get denatured, and also so you don't get ulcers in your duodenum. Okay, so that's important, right? We've got these two actions of secretin to calm things down. I want you to think about it as inhibiting acid. Now, the last kind of important cell we'll talk about on this page, I'll make this in green, and these duodenal cells, they release a hormone called cholecystokinin. It's a big word, so we're going to give ourselves some room to write this. Cholecystokinin, or CCK. Isn't that easier, calling it that? And it also has two jobs. One of its jobs is to also affect the pancreas, but instead of having the pancreas make the baking soda bicarbonate, it actually causes it to release all of its important digestive enzymes that can break down carbohydrates, proteins, and fats. So that's going to be one important job. And then the other thing that CCK does that's very important and notable here is it stimulates the gallbladder to release bile. And bile is an emulsifier that will break big fat globs down into little fat globs so that the enzymes from the pancreas can chemically digest it. And how these enzymes work all together is what we'll talk about next. Once the acidic chyme starts coming down into the duodenum, we are going to see the liver and the pancreas and the duodenum work together to finish chemical digestion. First of all, the liver is the important start point for bile. The liver makes bile, and then the gallbladder stores and concentrates that bile. I'll go ahead and color over here the gallbladder. Here's the gallbladder. And then this whole big structure is the liver. And little cells in the liver called hepatocytes work together to make bile, which I'm using green to represent this. And then the bile drains down away from those cells until it gets to what is called the common hepatic duct. So that's right here. Common hepatic, that word hepatic just means liver, duct. And that then from there it can go up into the cystic duct, which is this part right here, and be stored in the gallbladder. Then the hormone that we've already talked about, CCK, cholecystokinin, that's the one that will stem the release of the bile. And that bile then will drip down the common bile duct, which is this area right here. It kind of goes behind the stomach there. So, and then that will go, so this is the common bile duct. And then that bile passes through a little sphincter right here that is called the hepato, because that's liver, pancreatic, because it's also going to empty things from the pancreas, ampulla. And that word ampulla, when you see that, that means a widening. Little widening where then those chemicals, the bile, can empty into the duodenum. So it's able to come out into the duodenum by passing out this little bump inside the duodenum called the major duodenal papilla. The purpose of that bile is to do what's called emulsify lipids. That's kind of a big word, big fancy word, but all it means is that it will break big droplets of fat into little drops of fat. And then the enzyme called lipase will be able to work more effectively on it because it will have a bigger surface area. The pancreas doesn't make bile, it makes the enzymes that can break down chemically the foods that we eat. So those emulsified lipids that we talked about, they are going to get broken down by an enzyme called lipase. And whenever you see ACE on the end of something, you know it's an enzyme. And we're going to write lipids right here. And I'm going to actually add the word emulsified. So this has been broken down into little drops mechanically, and then will chemically be broken down by lipase into fatty acids and glycerol, because triglycerides have 3 fatty acid tails and a glycerol head. And I'll just represent those with these dots right here. Now imagine that the duodenum, which we have over here, I'll go ahead and outline it in purple. So it's just the first 10 or 12 inches of the small intestine. And it is lined with cells called enterocytes that have microvilli on them. So I'll draw it like this. And this is going to then be labeled an enterocyte. The word entero means intestine. So this is an enterocyte in the small intestine, and actually would be in the duodenum and probably jejunum as well. And I'll give it a little nucleus. So those fatty acids and glycerol are able to be absorbed right through these cells, and then they enter vessels of the lymphatic system called lacteals. And I'll draw them in yellow because they're not a blood vessel, they contain lymph. So we call this a lacteal. And it is a lymphatic vessel. And so the end result is that those fatty acids and glycerol are absorbed into the lacteals. And then from there, they get dumped into the bloodstream near the heart. So the heart gets first dibs on these fatty acids, which it loves to use to make ATP. Polypeptides can also be broken down in the duodenum. And rather than being full-on proteins, we can thank pepsin, because remember, pepsin is active in the stomach and it breaks large proteins down into polypeptides that then can be made even smaller by pancreatic enzymes into small peptides. Not yet down into amino acids, so they're not ready to be absorbed yet. And the enzymes that do this are called proteases. There are a variety of them. Probably the most famous is called trypsin. So I'll go ahead and write that one. And so then we have these small peptides. Not quite ready to be absorbed yet though. They need a little bit of help. They can't be absorbed straight into the lacteals like the fatty acids. Instead, they require the help of brush border enzymes that are in the small intestine. So we're going to call these brush border enzymes. And these are found on the enterocytes. and they are required to finish up the digestive processes for both proteins and, as we'll see, carbohydrates. I'm going to wall off the part about the liver up here. So let's draw an enterocyte right here, kind of at an angle like this. You'll see what I'm doing here in a second. We're putting the microvilli on it and give it a nucleus. And this is a cell that's lining your duodenum or your jejunum, so the small intestine. And there's always capillaries associated with these cells so that things can either pass through them and get absorbed, or like in the case of the fatty acids, there's also lacteals. There are enzymes, and I'll make these brush border enzymes orange. And these enzymes are capable of breaking the small peptides down into amino acids. So I'll write small peptides and then amino acids here. So those enzymes make that happen, those brush border enzymes. And now we have something that is small enough that can be absorbed through the enterocyte and enter the bloodstream. So see how the pancreas enzyme started it with proteases and the brush border enzymes of the duodenum finished it off. I'm going to take a moment to outline the pancreas in blue. I forgot to kind of draw your attention to it. And right here, this is the main pancreatic duct. And so the pancreas in these acinar cells here will make, so these are acinar cells that make the enzymes, and then those are able to come out that major duodenal papilla and enter the duodenum, where then they can start breaking down the proteins and the lipids and the carbohydrates. Acinar cells of the pancreas are where the enzymes are made. All right, back to work down here. So we're going to look now at carbohydrates and polysaccharides that are smaller now than starch are able to be broken down further. So we're going to say think saliva, because there is pancreatic amylase in your saliva as well. Polysaccharides are broken down into oligosaccharides, which are smaller by none other than amylase, which we've talked about before, but it's also made by the pancreas. So then it can drip, drip, drip out into the duodenum to help break down starches and other kinds of carbohydrates. But notice now these oligosaccharides, they are not yet broken down enough to be able to be absorbed into the bloodstream. So just like the proteins, they need a little bit of help from brush border enzymes. Let's go ahead and show how that works. Okay, let's draw an enterocyte at an angle so you can see what I want to show you. And we'll put the microvilli on there and give it a nucleus and draw a brush border enzyme right here like this. But this is going to be a brush border enzyme that is good at breaking down disaccharides like lactose, sucrose, or maltose. In fact, some examples of brush border enzymes that are important in the duodenum for carbohydrates are called lactase. You can guess what that breaks down. Sucrase. That breaks down sucrose, which is table sugar. And then maltase, which breaks down maltose, which is another kind of disaccharide. And then that is going to, so I'm going to go here and put that you have disaccharides, which are two put together, just a little bit smaller than oligosaccharides. So disaccharides are broken down into monosaccharides, glucose, fructose, or galactose. Those are the options there. And now they're finally small enough to be able to be absorbed into capillaries in the submucosa. Like that. So in both cases, whether it's amino acids or monosaccharides, they're going to be absorbed into capillaries. And that's, you can see that I've drawn those in red. And then same here, absorbed into capillaries. Now really remember that that is different, because if you're taking a traditional A&P class, you will probably get a question on your test, making sure you know that lipids, fatty acids, they get absorbed into lacteals, which are lymph vessels. These are capillaries. Another thing that the pancreas does under the influence of secretin is to add bicarbonate to the duodenum. So the pancreas also secretes bicarbonate, because remember, you've got that acidic chyme. And it has endocrine little circles, like islets of cells. So it also secretes hormones into the blood. We'll talk about those later. Insulin being the main one. But I'll put etc. since I'm running out of room. There's also glucagon and somatostatin. Here I want to show you the 4 main layers of the GI tract. They aren't always like exactly the same, whether it's from the stomach or the small intestine or the large intestine, but a lot of it is similar, and so we can have some main takeaways. What I'm drawing here first is the mucosa. These are lined with enterocytes, and this is a mucous membrane. So I'm going to put over here, number 1, first layer, is the mucosa. The mucosa is made up of simple columnar epithelial cells, and usually they have microvilli on them. So I'll draw these. You might remember that enterocytes will usually have microvilli. We'll write it like this. So these are going to sometimes have brush border enzymes on them, which are enzymes that help aid in the digestion, the chemical digestion of the food. So we'll put some have brush border enzymes to help out, because the pancreas has most of the enzymes, but we do need these brush border enzymes. So if someone has a disease like celiac, it can affect their ability of their brush border enzymes to be healthy and have enough of them. So this is going to be the inner layer where the nutrients need to be absorbed through the cells. And the next layer we're going to talk about is the submucosa, and that's going to be where those capillaries and lacteals are for absorption. So we have to have that absorption happen through the enterocytes though. And then a couple of other kinds of cells you find here are going to be goblet cells, for example. They got named because they have a very goblet-y look to them, and they make mucus. This is a mucous membrane, so that shouldn't be a surprise. And there are antibodies that line our mucous membrane, and these are called IgA or immunoglobulin A antibodies. And these are specially designed to protect mucous membranes. So we have these on all of our mucous membranes, and they are very plentiful in breast milk so that babies get to have their mucous membranes protected even when they're still little and pretty vulnerable. So we'll look at the submucosa next. I'm going to use green and make a line around. So now this is going to be the second layer as we're looking out. And we can put some capillaries in here into the submucosa layer, sub meaning under, so it's under the mucosa. And then also some lacteals. These are where the triglycerides, the fatty acids, will eventually get absorbed. Let's go ahead and add some brush border enzymes onto this mucosa lining so you remember that these are enterocytes that have these on them. And we can put some amino acids and some fatty acids and some monosaccharides. And these are the nutrients that then can be absorbed into, I don't want to do that, can be absorbed into the capillaries, or if it's a fatty acid, into the lacteals, if it's a monosaccharide, into a capillary. So we have different kinds of absorption there. Now I'm going to just color this in real faint so I don't go over anything. So this is the submucosa, under mucosa. So if you were to take a cross section of a piece of intestine, these are the layers you would find. So we'll do layer number 2, submucosa. This is where we find the blood vessels to absorb amino acids and monosaccharides, and the lacteals that can absorb fatty acids. You also find lots of mast cells here. These are a kind of white blood cell, and they're going to help, again, to defend your body against foodborne illness. Okay, then the next layer out is going to be a layer of muscle. This is one of the layers of the muscularis externa. This innermost layer is circular, so I'm going to draw it like this. This just represents circular, smooth muscle that can squeeze down on whatever food is in the lumen and help to move it along. So we call this the third layer, the muscularis externa. And you've got circular, which we just drew, and longitudinal is most typical. Now the stomach also has that oblique layer, but most of the GI tract just has the circular and longitudinal layers. So I'm going to draw another layer out here to represent the longitudinal layers. This one's a little harder to visualize because these muscle fibers would be coming at you. So the truth is that the best way to show it is like little circles that you would see. And these would be like fibers coming at you since it's a cross section. If it doesn't make perfect sense to you, really don't worry about it. What you need to know is that there are 2 layers. There's a circular layer and there's a longitudinal layer in most parts of the GI tract. Okay, so we've got circular and longitudinal. And the purpose of these is to perform peristalsis, which is going to move the foodstuffs along in a forward fashion. So I'm going to say moving it along. And then there's another kind of important contraction that occurs that's called segmentation. And this is when it kind of moves it forward and then backward and then forward and then backward. And the overall effect is an important mixing effect where the contents that are in the lumen are able to bump up against the walls of the mucosa over and over again. Okay, then we'll do the serosa. And I'm going to pull this back. Let's go ahead. The serosa is also known as the visceral peritoneum. So this is a membrane that surrounds, it's kind of glistening looking, and it surrounds the outside of each coil of intestine. And not only that, but it will often double over on itself to form what we call a mesentery, so that there is a pathway for blood vessels and nerves to get to and from all the different places inside of the peritoneum. It's like little highways, you could almost think of it. And this area, I don't really like how I drew this. Let me fix this. So this, first I guess I should label it for you, right? Okay, so this is the serosa. Also known as the visceral peritoneum. When you see it folded over like this, this is what we call a mesentery. And that's really important because it anchors all the coils in place. And it also is a path for vessels and nerves. And especially in the really prominent ones like the greater omentum and the lesser omentum, it's a great place to store fat. And so as people maybe gain weight, you'll notice that their greater omentum will be thicker and thicker. And the greater omentum is actually where someone has a lot of visceral fat. That's where their fat is stored. But we also would have blood vessels. So it's going to give a pathway for those blood vessels and nerves to come and go. We will wrap up our discussion of the digestive system with looking at the colon. The start of the colon actually has a valve that marks the end of the small intestine, and we call that valve the ileocecal valve. It's really a sphincter muscle, ileocecal valve. And it's going to prevent feces, which are forming in the colon, from going back into the small intestine. The first part of the large intestine is called the cecum, right here. And it is a blind pouch at the start of the colon. I'm going to put this right here. Now, hanging off of this is a very interesting structure known as the appendix. Most people have heard of this, right? The appendix, we believe, has a lot of immune functions, and it can hold a reservoir of good bacteria to repopulate after you've had bad diarrheal disease. So the appendix hangs here, and we believe that it serves immune functions. And if people get appendicitis, in my mind, it's a little bit like someone that is getting, swollen tonsils a lot. Your appendix is a lymph tissue, but it's just in your colon instead. And some people can have repeat and chronic appendicitis where it's inflamed chronically without perforating, meaning exploding out basically into the peritoneum. And usually they'll have it removed, but it can be a chronic problem too. Now, before I move on, I want to show you this smooth muscle that bunches the colon up into distinctive segments that are called haustra. So that's what that line is going to represent. It's the tinea coli. Let's put that right here. Tinea coli. And these are bands of smooth muscle that bunches the colon into segments. And we call those segments haustra, like little bands of sausage, basically. As the colon goes up, we call it the ascending colon. And then it turns the corner right here at what's called the hepatic flexure, because that's where the liver is. So this curve right here is called the hepatic flexure. Then the transverse colon goes across the top until it gets to the splenic flexure, which is this turn right here. The splenic flexure, where the spleen is. The large intestine is retroperitoneal, which means it's attached to the body wall, back body wall, and that's a good thing because we wouldn't want to get a twisted bowel, right? And so this helps to keep it from twisting up. It's kind of held in place. The mesenteries help with that too. Got the descending colon. And then the sigmoid colon we'll do in orange. See how it's considered to be kind of shaped like an S curve. And I'll make the rectum here pink. So this is the rectum. And then you can see that there's some muscles right here that are important anal sphincters. First is the internal anal sphincter. And this is involuntary. So if your bowel is full, then this smooth muscle is going to relax and you're going to feel very much that you need to have a bowel movement. And if you're not potty trained, you will. But as we become potty trained, then we also are able to voluntarily learn to control the skeletal muscle that is the external anal sphincter. And so this one will be able to hold it in until you have opportunity. So this is skeletal, which makes it voluntary. We have a similar arrangement for the urinary bladder with an internal and an external urinary and urethral sphincters that help us learn to be able to only void when we want to void. It's very important in reabsorbing water. If your large intestine is compromised, let's say you have a foodborne illness, then you might get very severe diarrhea to the point of death. So it's going to reabsorb water and electrolytes to help keep us having an appropriate blood pH and blood pressure. Then we also have billions of normal flora. That's just bacteria and some yeast, so microbes that live within us, more cells that live within us and on us than that make up our own body. And these normal flora have some important jobs. One, they actually protect us from being colonized by actual pathogens. So they're, for the most part, harmless, and they keep pathogens away. They also are able to help produce some vitamins for us, such as some vitamin K. And then we can absorb them into our bloodstream. And going along with protecting us from pathogens, they actually are important. They talk to our nervous system and help to modulate in a good way, usually, unless you have dysbiosis, modulate our immune responses. And if you have autoimmune disease, or now they're finding that autism, there might be an issue with the way that the normal flora communicate with our brain or the populations that we have there.